Science Journal for Kids and Teens presents What Can Fossils Tell Us About the Nervous System's Evolution? Adapted from the original peer-reviewed paper in the journal Geology, published on July 26, 2021. Research conducted by Russell D.C. Bicknell, John R. Patterson, and others from the Paleoscience Research Center at the University of New England in Australia. See the full list of authors and their affiliations in the accompanying PDF. Read by Miranda Wilson. Abstract. What can we learn from fossils? We can estimate the shape and size of an extinct animal. Anything else? Well, if soft tissues like the brain or muscles fossilize, it could tell us how the animal functioned or behaved. Unfortunately, soft tissue decomposes quickly after an animal dies. They aren't preserved as fossils very often. That's why we felt really lucky when we came across a fossil of an extinct horseshoe crab with a preserved central nervous system, or CNS. We discovered that the organization of the CNS in our fossil is the same as in horseshoe crabs living today. It hasn't changed in over 300 million years. We also figured out how our unique fossil might have formed. This could help others discover similar fossils in the future. Introduction. Have you ever wondered how you feel pain or understand what your eyes see? Your central nervous system, or CNS, controls and processes those and many other functions in your body. It also plays a role in your behavior and how you interact with the world around you. It is basically your brain telling you what to do. As you can imagine, the CNS in many animals is very complex. One way scientists can study the CNS is by looking at its morphology, or the way it is organized. This can provide a lot of information about the way the organism functions. Paleontologists can look at the CNS in fossils to see how extinct animals might have functioned or behaved. Unfortunately, the CNS is made of a soft material that decays quickly once an animal dies. This means there aren't very many fossils where paleontologists can see the CNS. With this gap in the fossil record, it's hard to understand how the CNS might have evolved in many animals. One group of animals where this is important is the arthropods. Arthropods are one of the most diverse groups of organisms, and they have been around for more than 500 million years. You have probably heard of some of the most popular arthropods, like insects, spiders, and crabs. We wanted to know what changes might have happened in the CNS of arthropods over time. Luckily, we found a fossil of an extinct horseshoe crab where we could see the CNS clearly. We took a closer look at the fossil to learn about the CNS in this extinct arthropod and to discover the possible taphonomic processes that led to the formation of this fossil. Horseshoe crabs living today are important marine arthropods. This is a photo of a baby Atlantic horseshoe crab, Limulus polyphemus. It is being held in a person's hand. You can see the helmet-like head, the abdomen, and then the telson, or tail. Methods. We looked at the fossil of a Euprupstanae horseshoe crab that is kept in the Yale Peabody Museum. The fossil was originally discovered near Mason Creek in Illinois, USA. The animal lived during the Carboniferous period, about 300 million years ago. We photographed the fossil. Then we used a scanning electron microscope to figure out the chemical composition of the minerals and rock making up the fossil. Results. We found that the CNS looked like a ring with 7 paired extensions. These extensions got longer toward the back of the fossil. Figure 1. This is the fossil of the Euprupstanae horseshoe crab we studied. The central nervous system, or CNS, is outlined by a white box on the fossil. The inset shows a redrawn version of the CNS. There is a scale directly above the inset indicating that the fossil was relatively small. We found that the CNS was preserved as a white mineral. The mineral contained a lot of aluminum and silicon, but not much iron, potassium, or magnesium. We identified it as kaolinite, a type of mineral. It has been seen in other Mason Creek fossils before. Discussion. Our find is exciting because it is the first time we have been able to examine the complete CNS of an extinct arthropod that lived in fresh water. The morphology of the CNS in our fossil is very similar to the CNS in horseshoe crabs living today. This means that the organization of the nervous system hasn't changed for at least 300 million years. This extinct horseshoe crab likely functioned and behaved in similar ways to horseshoe crabs alive today. Usually, soft tissues do not preserve well. This fossil is unique because the CNS is preserved in kaolinite, which is easy to see. We think this fossil must have formed in 3 steps. 1. The mineral matrix around the fossil formed very fast. The CNS was likely protected by a thick membrane. 2. The CNS eventually decayed and left a void. And 3. The void was filled in with kaolinite. Kaolinite is white in color and makes the CNS easy to see. We think there may be examples of soft tissues in other Mason Creek fossils, but they may be hard to recognize. Future studies should look at the color and contours of fossils to help find these preserved tissues. Conclusion. Fossils give us a snapshot of what happened in the past. Paleontologists can even figure out how organisms functioned and behaved by studying soft tissues like the CNS. It is important to look at fossils so we can understand how organisms have changed over time and how they might change in the future. If you want to see fossils, you can visit your local natural history museum, or you can take a trip to an area like Mason Creek to hunt for your own fossils. If you find something amazing, make sure to share it with your local museum or paleontologist. Thank you for listening to this recording. Visit our website, sciencejournalforkids.org, for more free science teaching resources.